Great. Well, it's great to be here, Davey. Thanks for having me down. It's the weather. We had pretty good weather in San Francisco, but I think you guys definitely have us beat on that count for sure. So what I'm going to talk about today is work that I've been doing over the last few years, and basically it's what I do and what I spend all my time thinking about is serological profiling. Basically, as an IT doc, as many of you are, my abiding interest is in applying this to understanding humoral responses to infections. So why bother with serological profiling? Hopefully this is not a hard sell to the group here that antibodies are key mediators of host immunity. They directly neutralize pathogens and toxins. They can promote phagocytosis through opsonization. They can activate complements. And then finally, an existence of a serological response is an indicator of exposure. So antibodies are both important for host defense, but they're also really key. Measuring antibody responses is a useful way to know what people have been exposed to. So it actually has diagnostic applications as well. So there are a lot of different ways that we use to study antibodies. Obviously, functional assays are sort of in many ways remain the gold standard. And so looking at things like antibody neutralization of viruses is a key way to measure antibody function. IG-Seq, so a more next generation high-tech-y approach where you're isolating B cells. Yeah, classic IG-Seq, you're talking bulk B cell sequencing. Obviously, now with 10x, you can actually sequence repertoires. You can sequence single cells, and so that tells you something about the variability of host responses to infections. And then there's what I do, which is phage immunoprecipitation sequencing. And so this is basically the marriage of phage display, a decades-old technique with next-gen sequencing. I like to always start off with a quick reminder of what bacteriophage are. So bacteriophage are viruses that infect bacteria, and they can cause lysis, or they can enter or they can enter what's called a lysogenic cycle, which doesn't involve lysis of the cell. Phage come in many different shapes and forms, like other viruses, there can be variants in the structure of they can have an envelope, they can be non-enveloped, their nucleic acid can be RNA, can be DNA, can be double-stranded RNA. So not very different than the viruses than eukaryotic viruses. And one of the key features of bacteriophage that has really made them a workhorse tool for decades is that they're relatively easy to manipulate genetically. So probably the two most commonly used bacteriophage are M13, which is kind of the sort of the original phage vector, and it was that's what people used to display fabs and large proteins. It has been a workhorse in the pharmaceutical industry for decades, particularly for monoclonal antibody discovery. And then T7 phage. And so T7 is I actually work with both, but T7 is what we're really, the work that I'm going to be talking about today is really built on. And I mentioned that the phage have different, they can cause cell lysis, or they can enter enter a lysogenic life cycle. T7 is a lytic phage. So just to sort of emphasize the value and the importance of this particular of phage, there was the 2018 Nobel Prize in Chemistry was actually awarded for phage display. So the work of Arnold, Smith, and Winter, and literally the prize was awarded for this phage display of peptides in antibodies. So it's a really important tool. And so the awarding of the Nobel was kind of a nice capstone and recognition of that fact. So what I work on is phage immunoprecipitation sequencing, or PhIP-Seq. And what this really is at its core is a very effective and rapid way to discover antibodies that target linear epitopes. And at the core, I mentioned the genetic pliability of phage, and that's really what makes PhIP-Seq, or really any phage display technology, work. So in the case of PhIP-Seq, what one does is, one starts with a proteome of interest. So that proteome could be all human viruses. It could be tuberculosis. It could be flaviviruses. These are things I'm mentioning because I will be talking about them in short order. One takes that proteome, decomposes all of the proteins into a series of overlapping peptides, designs nucleic acid sequences that code for those peptides, and then clones them in parallel into a phage display vector. So the end result of this is that you have basically a single peptide displayed on every unique phage, for which you know the code, because you designed the nucleic acid sequence that codes for that particular peptide. So just walking through how the experimental procedure works is, once one makes the phage library, which is basically a mixture of phage, each displaying a different peptide with a specific nucleic acid sequence that codes for that peptide that you designed. You take that mixture of phage, you incubate it with a patient sample. This really can be any antibody containing sample. Typically what I've worked with is serum and plasma, but it really, in theory, could be done with anything. I know some folks have done it with urine. I have some colleagues that do it with CSF. Anything that has antibody in it is fair game. So the antibody containing sample is incubated with the phage library. The antibodies are captured on protein A by using protein A and G that's been conjugated to magnetic beads. And essentially what ends up happening is antibodies that recognize peptides that are in one's phage library are retained, and the phage that are not bound to antibody are washed away. And ultimately, what one is able to do then is to take that, you sequence it, and what you get is, as output is a read table of peptide and the number of reads for that peptide. So if somebody has a lot of antibody to a peptide, a specific peptide, that peptide is going to be represented with a lot of read counts. So our work has originally started out in the autoimmune world, and I do a little bit of autoimmune work myself. But I'm just going to show a very quick example of the power of this approach as applied to autoimmunity, which then obviously extends to infectious diseases as well. This is a case that they saw at the UCSF of a 15-year-old girl who was diagnosed with anti-Q antibody encephalitis and seizures. So she had come back to the Bay Area and was experiencing episodes of nausea and vomiting, difficulty with word finding. And a few days later, her mom found her having a generalized seizure. She got hospitalized for 2 weeks, and had a little bit of a pleocytosis, an elevated protein in her CSF, and a markedly elevated anti-Q antibody titer. CSF from this patient was run using a PhIP-Seq library that represents the entire human proteome, and identified these fragments. And basically, this QD gene, this ELAV4 gene, was what this patient's antibodies targeted. And what you can see is that by using this peptide-based approach, you basically can get a really nice coverage map of where the reactivity is. And so Didn't necessarily, so it was actually a really nice way to figure out what was really going on, what the underlying biology was in the case of this patient. So I had heard a lot about this coming to the RECE lab meetings, and I got really interested in the idea of applying PhIP-Seq infectious diseases. But I'm going to talk about today are three relatively short stories, because I don't want to, and just my goal here is to really give you a sense of the power of this approach and what can be done with it. So the first story I'm going to talk to you about is about dengue virus, and then that was really where my PhIP-Seq journey began, and kind of the original project. Then, as is true for probably many of us, I veered a little bit into SARS because there was really, you see it in 2020, things pretty much got shut down for anybody that wasn't doing SARS work. And then we thought, as I'll talk about in a little bit, that we might have a useful way to deploy our technology to do something really valuable for SARS. And then the last story I'm going to talk about is, unlike the first two, something that isn't yet published, some preliminary data that's looking very promising on some work I've been doing with tuberculosis. So, starting with dengue. Dengue, for anybody who's traveled certainly in South America or Asia or certain parts of Africa, dengue is no stranger. It continues to cause substantial morbidity. And as everyone here knows, it's a vector-borne illness carried by the Aedes aegypti mosquito. And can cause pretty profound symptoms. And then obviously, because there's 4 different serotypes of dengue, people who are infected by different serotypes can go on to develop a really bad complication, which is dengue hemorrhagic fever, more common in children, certainly than in adults, as everybody here knows. At the same time, there are several dengue vaccine candidates that are in late stage or finished clinical trials, one of which has already been approved. One of the interesting questions that we had and that I had is that, we know that people who are serially infected by different serotypes of dengue subsequently have cross protection against infection with a third serotype. So if you serially get infected with dengue 1 and then dengue 2, and then you're exposed to dengue 3 or 4, you have protection against those against those different serotypes that you've never seen. And so that sort of has always raised the possibility that maybe it might be possible to develop a vaccine. A vaccination and natural infection are really different entities. In the case of a natural infection, somebody's getting bitten by a mosquito twice, and with 2 different strain serotypes of dengue. This results in what we think is lifelong immunity. There are, of course, as always, exceptions to this. Whereas in the case of vaccination, what we're talking about is somebody getting injected with all 4, with basically attenuated versions of dengue simultaneously. So there's a lot that's different here. Apart from the fact that you're getting all 4 viruses at once, there's also the whole issue of when you're getting bitten by a mosquito, you're not just getting the virus, you're getting all those mosquito salivary proteins, some of which can be immunogenic. It may affect the response that you have. And so we were really interested in getting at and trying to understand, are there really any differences between what are the differences between natural infection and versus unnatural infection or injection? And so we were able to identify, we had some collaborators who really had the perfect set of samples to assess this. And we thought that PhIP-Seq could be a really useful way to dig into this question. So collaborators of ours at Walter Reed had infected rhesus macaques with dengue, dengue 1 by both mosquito and by subcutaneous injection. And then even better, they serially followed the macaques. So we had baseline pre-infection and collected serum at multiple time points. So obviously at baseline pre-infection, but then at day 7, day 35, day 90, and up to a year post-infection. And so this was really, to us, the ideal design for trying to get at this issue of what are the differences between being infected by a mosquito versus getting a subcutaneous injection, and really, what effect does that have on immune profile? So what I'm showing you now are just figures from a paper, our paper that we published on this, and this is what you get with PhIP-Seq. So essentially, what I was able to do is generate coverage maps of reactivity across the entire dengue proteome. So this is 7 days post-infection. As you might imagine, there isn't that much signal. It's still pretty early in the stage. Probably measuring some IgM here, maybe a little bit of IgG. By 35 days post-infection, however, you can see pretty substantial reactivity, and in the regions where you would expect it to be. So we know that the envelope protein, which is the main surface antigen for flaviviruses, is a crucial target of host antibodies. And similarly, the NS1 protein, which has a lot of different functions, is also known to be a pretty important target in flavivirus infections. And so, unsurprisingly, that's where the signal is strongest. What I'm showing here is there's a region, I've highlighted a region in domain 3 of the envelope protein, which consists of 3 separate domains. And you'll see in a second why I've highlighted that. So 35 days post-infection, we saw pretty broad reactivity, mostly concentrated in the envelope and the NS1 proteins. By 90 days post-infection, those signals are still maintained. Again, mostly in the envelope and in the NS1 protein. But at a year post-infection, what you can see is while that initial hump of blue and red, where the blue is basically subcutaneous infection and the red is mosquito infection, there's no difference, but there are multiple other regions where reactivity is a long-lasting effect on what the shape of the immune profile is over time. So that gray region was the one that I was particularly interested in, and then I basically was able to validate that orthogonally using an ELISA, where I basically cloned, I generated that entire protein fragment where there was prolonged reactivity in mosquito-infected, but not in subcutaneously infected mice, and was essentially able to recapitulate the result that I observed by PhIP-Seq using an orthogonal assay. So in this case, ELISA. And I think the one thing I want to really emphasize is that you don't just get a reactivity now. So the way the flavivirus library that I designed to use this experiment to assay these samples was really, we were kind of trying to do a couple of different things. And a big part of it was designs tiled. So the consecutive peptides were tiled every 8 amino acids. And that was really to allow us to do some epitope mapping. And then the second design decision was to incorporate all of the genetic variability of dengue. So instead of just using a reference sequence, I actually used all of the available sequences from GenBank for dengue, with the idea that we really wanted to try to capture the variances in binding. And so what this resulted in was a really rich set of data. And at some level, what you get from a single assay is using this kind of a library is a mutational scan. But then one of the other things that we've kind of routinely seen, and that's shown at the bottom here, is that it turns out that a lot of these reactive regions that I identified in this case, and this is true for other work from our group as well, actually overlap with reported T cell epitopes. I don't know what the underlying biology of that is, but again, it suggests that using this kind of an approach, that we're not just learning something about antibody reactivity, it might actually be telling us a little bit more about the shape of the immune response. So just to kind of quickly summarize this dengue story and how and why I thought why PhIP-Seq was really valuable in this case. The core message here is that how the virus is delivered really can affect the shape of the immune profile over time. And so to me, what that potentially suggests, and I'm not saying that I have any proof of this, but the potential implication of this is that one wonders whether this might say something about the duration of vaccine-induced immunity for vector-borne illnesses, as compared to sort of what happens in the case of native infection. And so, there are a lot of different directions that we were really thinking about pushing this work. One was to look at primary versus secondary infection. The other, obviously, is it's one thing to identify the regions of reactivity, but really the next stage is to identify the specific antibodies that bind to these regions, and then try to do some functional assays. Are these potentially neutralizing? Are they really non-neutralizing? And just kind of understand biology a little better. And then, one of the other things we were really interested in was like, okay, we've done this in macaques. Can we look at natural infection versus vaccination in humans? And so I built a relationship with collaborators in Brazil, where there's a large ongoing dengue vaccine trial using the NIH vaccine. What I really want to just emphasize is that, to me, what these results showed was that PhIP-Seq is a really sensitive tool for measuring differences in immune response. And with the right cohort and the right study design and the right library, you can really do some interesting work and get into some interesting biology. So I was all geared up to continue pushing this work forward. Our collaborators at Walter Reed actually had wonderful other samples that we could use, and then SARS hit, and pretty much everything got shut down, and nobody was allowed in the lab. And like for many of us, this was a really big challenge, but it was also, in some ways, a bit of an opportunity. So as folks probably remember that diagnostic testing was a real problem in the early stages of the pandemic. While PCR assays came up relatively quickly, they're slow, and obviously are best for acute infection. And at that time, there really were not a lot of serological tests available, and so we felt that there was there's always a role for serological diagnostics. They're generally easy to do, especially if they're implemented as a lateral flow assay, and at the very least, they tell you about exposure versus non-exposure, which in the early stages of the pandemic was actually a pretty important question. So we thought that what we were doing with phage could directly be applicable to this problem. And so what we developed was something we ended up calling a rescan. And what this rescan is essentially a single workflow for antigen identification and assay development. So this is also published work. Essentially the idea is we take a set of samples from people who are sera from persons who are exposed to COVID. You pull them, you use those to pan a phage library to enrich for antigens that are seen across multiple individuals. And then one takes that selected set of gauge, does a plaque assay, and then picks plaques basic in an unbiased way. So you don't know what you're actually finding. You just know that they've been enriched by the sera of people who you know have COVID. And you isolate each of those clones individually, and then we were able to print them onto nitrocellular, basically use an acoustic liquid handler, so an echo, to print them onto a nitrocellulose membrane, and then counterstain them with COVID-19 sera, and basically use that to identify sort of an optimal antigen set for a diagnostic antigen for a diagnostic test for COVID. And so I just want to emphasize how quickly we were able to do this. So COVID really, started to impact all of us by the end of February. And the sequence was up. And so I downloaded the sequence at the beginning of March 2020. And I'd already written a pipeline, basically, that makes it very easy for me to spin up PhIP-Seq libraries. And so that second week of March, I designed a COVID library, I cloned it and sequenced it. And by April of 2020, we'd already run our samples. And by the first week of May, we had a set of biomarkers. So, it was about 8 weeks. I'm not going to lie, it was a very intense 8 weeks. We went from nothing to having a set of diagnostic biomarkers. So the first, just walking you through this, and these are just figures from our paper. The first step, as I mentioned, is to really enrich for peptides found in persons with SARS-CoV-2. And what I'm showing you here is the heat map of reactivity across the SARS-CoV-2 proteome for a bunch of positive control for positive controls. And what you can see is primarily this reactivity, unsurprisingly, is in the nucleoprotein and then the spike proteins. So very consistent with what had previously been reported for SARS-1, and what's also been subsequently reported for SARS-2. So as I mentioned, once we take this pool, we take a sera from a bunch of people who we know had COVID, who are PCR positive, and these are for the most part convalescent sera within 4 weeks of the original diagnosis. We use that mixture of antibodies from those people to enrich for COVID antigens. We plate the bacteriophage expressing those antigens onto basically the plaque assay, so onto an agarose plate, onto a lawn of the E. coli. That generates plaques. Then we counterstain that with sera. And the whole idea here is to basically, the PhIP-Seq approach is very sensitive, but what you want to identify are antigens that are going to work in a downstream assay, and that's what the counterstaining gives us. So plaques that light up on the counterstaining are things that are going to ultimately be useful in a downstream diagnostic assay. So once those plaques have been selected, what we do is we print them, as I mentioned, on nitrocellulose membranes using an acoustic liquid handler. And essentially what you get is a pattern of reactivity to the different plaques, which varies from patient to patient. And this is just kind of a representation of what ultimately ended up being the 9 peptides that we really were able to capture the greatest number of people with COVID, and sort of, the main take-home point here from this figure is that there really is just a lot of variability. And again, this is a total of 9 peptide antigens that we were able to identify. So these were all things in spike and in the nucleoprotein. And essentially what I'm showing you here is another figure from our paper, where just showing you where the peptides lined up with the reactivity. And I think one of the things that I always like to point out is that, there isn't, what we're displaying are linear peptides. So unsurprisingly, you don't pick up a lot of reactivity against the RBD, which we know is a conformational epitope. But you get just blazing reactivity against the carboxy terminus of spike, and then the nuclear protein also gets pretty substantial reactivity. And then it's below here. What I'm showing you is the degree of conservation across the spike and the nuclear proteins. And then we actually ran these samples on using both a pan-viral library and the SARS-CoV-2 specific library that I designed. And essentially the main point here, this final panel of the figure, is that it just shows you a motif that presumably the antibodies were binding for at least one of the peptides. So the last stage, once you have these antigens in hand, is to deploy them as an assay. And so what we ended up doing was figuring out that we could conjugate bacteriophage directly to Luminex feeds, and we implemented it as a Luminex assay. And then we tested the performance on another set of independent samples, about 80 samples, and had pretty good performance characteristics. It wasn't the sensitivity of 91% and a specificity of 89%. But literally at the time we were beginning to finish this up, there was suddenly an explosion in the number of diagnostic assays that were available. Abbott ID Now came out and so on. And so we didn't ultimately end up pushing this further. But it is, to us, a really valuable proof of concept of how easy it is to do this using this approach. So for the next pandemic or for other potential, cases where there's a lack of a serological diagnostic, certainly an approach that can be used. So our SARS work continues. I've done a couple of collaborations with the folks at UCSF using the same tools, and this was a collaboration with Joe Sabatino, who's a neurologist, and what they were looking at was vaccination in people with and without MS, and those with MS on different immunosuppressive regimens. And what you can see is that that in people with MS but who were not on treatment, that their profiles look very similar to those of healthy controls. But if you use B-cell depleting therapies like Rituxan, that you completely ablate the signals. So both the profile and the underlying biology is really aligned well in this case. To me, one of the more interesting observations that came out of this is that natural infection and mRNA vaccination, so all of the people in Joe's study got either Pfizer or Moderna, and there really weren't any substantial differences by vaccine type, but that the signals are very different in natural infection versus vaccination. And so what you can see here is that while in the mRNA vaccination group, you get this huge peak here and like very large peaks adjacent to the RBD. That's less commonly seen in natural infections. You get sort of these more carboxy terminal peaks. And so this to me is just kind of another indication that there may be even more information in these kinds of data than we realize. Like I think I could probably, even sort of a large enough data set, use machine learning and pretty easily distinguish natural infection versus vaccination using PhIP-Seq data. And then another collaboration, again, continuing in the SARS world that I've done is with Mary Prahl and PhD at UCSF, and her and Stephanie Goff, who's an OBGYN. Basically, what they have a cohort of mother-infant dyads, and we're interested in looking at differences in maternal antibody transfers in 50. And essentially the main point here is that there really isn't, while there's variability between hosts and between that the mother and infant signals are generally, as one might expect, happily pretty consistent. So to summarize what the work we've done using PhIP-Seq and its application to SARS, what I really want to emphasize is what I think we developed is a scalable approach for serological assay development. Just starting with the genomic sequence, you can go. It's possible to develop a serological assay. So I think this is something that's applicable to many pathogens. And I think one of the things I always like to emphasize is that phage are a limitless and low-cost source of protein. I think the fact that we can detect differences between natural and vaccine-induced immunity suggests that there's far more that we could be doing with this approach. Again, with the right cohorts and sort of the right study questions. And then, really last but not least, PhIP-Seq data to me is both actionable and immunologically meaningful. Obviously I'm biased. This is what I work on, but I think it's a great technique, and I think there's just so much more we can do with it. So the last story that I'm going to talk about is in relatively preliminary stages, and I'm going to kind of breeze through this just to make sure that I leave enough some time for questions at the end. Tuberculosis is something that I've been interested in since I was an IT fellow. It was actually what I was originally supposed to work on as a fellow, and for a variety of reasons did not end up happening. It remains not just one of the top causes of infectious mortality worldwide, but really one of the top causes of death, period, which is pretty remarkable for a disease for which we've had treatment that for garden variety TB is effective, and we've had it decades. But is there anyone who's worked on TB or taken care of patients with TB knows diagnosis is really challenging. It can present in many different ways. Screening for TB is mostly driven by pulmonary TB and is driven by clinical symptoms such as cough. And while we do have diagnostic assays, AFP stains are fast, but they're not that sensitive because obviously, as everybody knows, people can have smear negative TB. Gene expert is also pretty fast, also not perfect. And then, ultimately it's culture that remains the gold standard, but it's slow. Mycobacteria are slow growing. And so this remains a conundrum, particularly in endemic, in regions where TB is endemic. What does a delay in diagnosis mean? It really does have consequences because, as people get started on treatment empirically, people get started on treatment empirically here too. I used to attend a TB clinic in San Francisco, and if the story was good and we were still waiting on culture, you start people on treatment because of the public health risk. But it's for drugs, and these are not things that are entirely innocuous on their own. So serology has traditionally done really poorly for TB. There are lots of serological diagnostic assays that are used in the endemic setting, but none of them have good performance characteristics. And people have looked at proteome-wide profiling for TB. This is mostly done by protein array, which involves expression of the protein of TB proteins in either E. coli or yeast, but many MTB proteins are actually really difficult to express. So my question was, could we do better using PhIP-Seq, sort of coming fresh off of this experience with SARS-CoV-2? So I basically ended up designing a tuberculosis PhIP-Seq library based off of the H37RB lab, commonly used strain of TB. And it's about 100,000 peptides, and much like my flavivirus library that I used for the dengue work I earlier discussed, it's relatively densely tiled. Again, this is just another little cartoon explaining how the whole PhIP-Seq process works. So I was able to, with a collaborator from UCSF, tap into a cohort that was a prospective cohort of inpatients with suspected TB. So everybody, and the key thing here was that everybody got sputum culture obtained in all patients. Sputum culture was obtained in all patients at baseline, so at study entry. And so we had ground truth about who did and did not have TB. And so this is really a great setup for trying to see if we can come up with a triage test, not an outpatient triage test, which is, of course, the ultimate gold standard, but a triage test nonetheless. This is basically a description. In the end, I ended up having about 129, 130 serum samples to work with. And so basically, I took the set of samples. These were people with and without HIV, and then with and without TB proven by culture. And so I ran these samples on my PhIP-Seq library. And this is work that I'm currently in the process of putting together to publish. And what we were able to find is that we can separate disease state by TB proteome reactivity. Probably, and I think that this figure actually sort of is instructive because it sort of explains one of the big difficulties in TB, which is maybe one of the reasons that serological testing may not perform that well is that probably a lot of the NTB proteome is reactive in people who do not have TB. And so there's one of the things I did to make this particular analysis extra conservative was to constrain it to overlapping peptides. So I didn't consider anything a hit if I didn't have like 2 overlapping peptides in a given individual. And using this approach, what we found is about 70% of the NTB proteome was reactive. So 2,725 different proteins, but only 25% of these. So the things that are in the overlapping regions of the and non-overlapping regions of the yellow and the blue circles here. These are proteins that are extremely specific to people from an endemic TB region. So either with active TB or no active TB. So these pretty much everybody was latently infected in this cohort, as you might imagine. This is a cohort of patients from Uganda. But 25% of the MTB proteome was seen, reactivity against 25% was seen only in people from endemic regions and never in non-endemic controls. And so just to emphasize that point, this is a heat map of kind of that subset of proteins that you only see in endemic TB, people from endemic regions. So essentially no signal at all. And then you start to see sort of pretty variable dispersed signal. I could cluster this, and there are other ways to show these data, but essentially what I'm showing you is reactivity across all of these proteins, from 5' to 3' across the NTB chromosome. Non-active TB, again, you can't really, it's hard to discern what the differences in the patterns are, but I think the next figure I'm going to show you will get at that. But bottom line, reactivity in endemic, there's about 977 proteins that use reactivity in people from endemic regions that you never see in non-endemic. So these are things that maybe might have utility for serological diagnostic triage assay. So, if we then turn to focus on which of these proteins are immunodominant, really the point I want to drive here is that active TB and non-active TB actually have slightly different patterns. So it's not the same proteins that are immunodominant. And here immunodominance is defined as present in at least 10%. So greater than or equal to 10% of hosts. So the patterns are distinct. And again, you don't see anything in the non-endemic controls. And so really to me, the question is, can anti-TB, MTB activity distinguish active TB from a non-active TB? Is this something that is potentially actionable in an assay? And so I think my sense is that we can actually separate disease state by PhIP-Seq profile. So if I just take that set of immunodominant proteins and I say, okay, I want reactivity against any of the peptides that are present in the immunodominant proteins, and I cluster, I don't really effectively separate active TB and no active TB. If I instead focus on I zoom out and go at the zoom in and go to the peptide level, and I say, okay, I'm just going to focus on immunodominant peptides. What you can see is that I get almost complete separation of persons with active TB from those without active TB. And so 52 of 54 active TB cases, so 96% really clustered together. And then all of the not the people without TB also clustered together. So, you know, pretty promising data. Now, obviously, for a serological assay, what we really want is to try to get down the number of features. And so when I start doing that, you know, I can get it down to as little as 120 peptides, and which span 84 different works. And the performance is still pretty darn good. If I push it a little further, then the performance starts to degrade. But even in this case, the thing I would note is that the people without TB are still sort of are still separated from the people, for the most part, from the people with TB. So pretty good specificity in this case. So we're pretty excited about this. I mean, obviously, there's a lot of work to do in terms of pushing this forward and rigorously testing it. But I'd say the initial returns are interesting. Thinking a little bit more about the biology, what's interesting to me is that almost 50% of these immunodominant peptides and the proteins that are specific to people with active TB or no active TB, but people who are from an endemic region, is that we really are targeting things that kind of make biological sense. So cell wall, so essentially surface proteins. These PE and PPE proteins are actually, nobody really knows what their function is, but they're thought to be, they're about 7 to 8% of the TB genome. And so similar to other bacterial pathogens, they're thought they might be involved in immune evasion. And so the fact that they're so heavily targeted is potentially quite interesting. And then one of the biggest categories is sort of conserved hypothetical proteins. So there's all these hypothetical proteins in the MTB genome that it turns out that probably are being made because we are able to measure signals against them. So just to summarize, the TB work, which is ongoing and hopefully will be coming out the door relatively soon. Much of the MTB program is reactive using this approach, but only 25% is specific to persons with active or not active TB. You know, I think one point I want to emphasize is that for diagnostic purposes, that peptides may actually perform better than whole proteins, and that the different performance of the clustering, I think, really sort of drives home that point. One thing I really believe in is that I think we need to really, for serological diagnostics, expand our minds and not be thinking about just small sets of antigens. Large host responses are highly variable. And so I think large antigen panels are going to capture that heterogeneity and may ultimately lead to better diagnostic performance. And there are ways to make that actionable. And frankly, this platform is a great way to do so. And so, and last but not least, I think the unbiased profiling approach it basically identifies targets that are telling us something about those pathogen interaction. So I hope after hearing what I've had to say today, you agree with me that PhIP-Seq is a powerful platform for looking at serological responses to infectious diseases. I think there are lots of different applications and that I've already kind of mentioned. Diagnostics, vaccine antigen discovery, treatment response monitoring, and then a last one is serological vigilance, which I didn't even really get into at all today. Bottom line is I really think we're just scratching the surface of what's possible using this approach. And then last but not least, I just want to give a big shout out to Joe DeRisi, who's my boss, and is the guy who got me really excited about science again, and it's just been just a wonderful guy to work with, and we just have a fantastic community. Hannah Ritalik was a grad student who was working with me on the PhIP work to start. Michael Wilson is an ongoing close collaborator. And then some of these other folks are the folks that I talk to a lot, all the time. And then my TV collaborators that did Kadamachi, Devin Dragon out of Babak Jari. That's my daughter, and a picture from a camping trip I did up in Sequoia, Kings Valley, a solo backpacking trip a few years ago. Thank you. Happy to take any questions.
You have two questions in the